Good evening. My name is Jim Wilson, and I want to welcome you to one of several webinars on the science, policy, politics, and medicine of the COVID-19 epidemic. I am currently a faculty member at the Perelman School of Medicine at the University of Pennsylvania, and I'm also a proud alum of Albion College. This webinar series is being sponsored by the Lisa and James Institute of Medicine at Albion College. So before we get started, I'd just like to review the format for the next hour. After our featured speaker makes some introductory comments, I will then welcome several students that are currently at Albion, who will then lead a discussion with our distinguished speaker. I'd also want to remind the audience that there is the ability to ask questions in the Q&A function, and I'll definitely be watching for that and try to get your questions answered. So we're really looking forward to tonight, incredibly timely talk. It's a pleasure to welcome a friend of mine, Stéphane Bancel, who's currently Chief Executive Officer of Moderna, which is a novel technology and product development company that's focused on a platform of mRNA therapeutics, and Stéphane will tell us about that. He was trained as an engineer both in Paris, where he was born, and University of Minnesota, and then received an MBA at Harvard University. He then took on several leadership positions in the pharmaceutical industry, and then joined Moderna soon after it was founded in 2011, and took it public in a very successful IPO in 2018. As we all know, Stéphane has jumped in to try to, and his company, to try to develop a solution to this awful pandemic, and it's a pleasure to have him here. And I'll pass it over to Stéphane. I'll make a few introductory comments. Stéphane.
Well, thank you, Jim, for the kind words, and good evening, everybody. Thank you for having me tonight. It's always a pleasure to go back and talk to students and to engage with the next generation of scientists. It's very exciting for me. So what I propose to do is to talk briefly about Moderna, then talk a bit about the COVID vaccine that our team has been working on, and then I will talk a bit about some of the things we're hoping to do for the future so that such a pandemic cannot happen with such a magnitude in the future, and then be happy to turn back to Jim to have a discussion and a dialogue with you guys. So Moderna was indeed started in 2010. I joined in 2011. We had one scientist at the company. And the whole idea that the founding team had, actually the whole question that the team had is, what if messenger RNA could be a medicine? And I have to say that when I was first presented with the idea myself, within 5 seconds, my reaction to them were, you guys are crazy. mRNA will never be a drug. It's too unstable a molecule. It's highly immunogenic. Those two things do not go well with making a medicine. But they were able to make me wonder, what if we could figure out the science, the manufacturing process to make a safe and efficacious mRNA? What would it mean for patients? And this is where I think I got really excited, because if mRNA could be a medicine, you could make medicines that could not be done using small molecule or large molecule, i.e. protein or recombinant protein, especially in the intracellular or transmembrane space. And I knew that around 2/3 of the protein coded in human DNA are coding for proteins that are intracellular or transmembrane, so they are not druggable using existing technologies. The second piece that was very exciting was the notion that mRNA is actually an information molecule. Because as most of you, if not all of you know, mRNA is made of 4 nucleotides. And so if you were to figure out how to make the first medicine, by changing the sequence, you could very quickly figure out how to make another medicine. And that, to me, who had kind of spent the early years of my career either making recombinant at the bench in grad school or at commercial scale in big pharma was really attractive. The notion that you did not have to invent everything every time again for every drug, which is what the pharma industry has been doing for 150 years. And so that information nature of mRNA molecules, I thought was going to increase the probability of technical success of drugs. As most of you know, very few drugs that leaves the labs make it to market to help patients. But because mRNA is information, it made no scientific sense that if one drug was going to work, let's say for a rare disease in the liver, you could not very quickly make another one based on the sequence of human DNA to go after another disease with exactly the same technology. The third piece that excited me was speed. Drug development and drug discovery is a very slow process, and every day patients are hurting, their families are hurting, people are dying. And so because mRNA is a platform, because mRNA is an information molecule, what I thought is that if you were to invest in robotics, in process engineering, in robotics, and you could build the manufacturing process correctly and all the manufacturing plant, then you should be able to go very fast to iterate and to generate a lot of molecule for preclinical research in the lab or molecule for clinical trials. That was exciting. So when you think about it and you step back, which is what I did that evening when they talked to me about the concept of Moderna, was like, well, if we could make this company work, we could make medicines that you cannot make with other technologies. If you were able to make one work, it made no scientific sense that you could make a lot of other molecules work, and you could go much faster. So that those three things together were extremely exciting. So I decided to resign from a company I was running, which was a big, 6,000 people company, to be employee number 2 at Moderna. From the get-go, again, because of how the science works, and it made no sense this will be a one-drug company, we said it's either going to be zero because we fail, because we don't raise enough money, because the science doesn't work, or if one makes it to approval, it's going to be a company making a lot of impact on patients with a lot of different medicines. And so from the get-go, we build this company assuming success, because as you can appreciate as an entrepreneur, you don't get up in the morning and say, okay, the company is going to fail, so I'm going to do nothing. So it was actually very easy imperative for us, which is we built assuming a big success, and we invested in science at extremely high level of investments, because again, we said this is going to be a big platform, so we need to really understand how the science works. We need to invest in manufacturing. We built a very large plant in Massachusetts before we had our first product approved, because it was an important enabler to do the science correctly, to do the clinical trials correctly. And as we thought about the company, assuming a big success and playing the movie backward to us in the early days with 2 employees, we asked ourselves the questions, what could derail us? And very quickly, we zoomed in on 4 big risks that I became obsessed about, because I thought if I could reduce those risks, the company should be able to be successful. And so those 4 risks that we focused on were technology risk. So of course, when you go after a new technology, you have a lot of technology risk because you don't understand the technology, you don't know what you don't know. Biology risk, which is how well diseases are understood or not understood. Just think about HIV as an example. It is still to this day a disease that is not fully understood. Think about cancer, Alzheimer. The third risk was execution risk, which is if we had the best plans in the world to reduce technology risk and biology risk, could we make it happen? Could we hire the right people? Could we organize in the right way to be able to execute? And the fourth risk, which is present in every startup, is financing risk. Can you get enough money to do the work correctly? Or are you going to run out of money before you have enough success, either to get to cash flow profitability from selling your products, or to a point where investors think you made enough progress that you get a new financing? And those are the things we focused on. And so at the end of last year, the company had around 22 medicines in development 40 plus program in research. We have our own plant in Massachusetts. And we started the year, of course, focusing on how we're going to do more products, how we're going to do all those phase 2s and 3s, and how we're going to continue to develop the pipeline to hopefully get to a product launch. And then in the first few days of January, I read that there was a new respiratory infectious agents causing pneumonia-like disease in China. And as soon as I read that, I basically emailed the team of Dr. Tony Fauci at the NIH that we have been partnering on for several years on different infectious disease vaccine candidates. And I asked them, hey guys, you heard about this thing in China? Is it a virus or bacteria? Because we didn't know. And they're like, we don't know. We'll let you know as soon as we know. And a day or two after, they emailed me, it's not a bacteria, it's a virus. And it is not flu. I mean, all of us in the field were always worried about a new strain of flu, like you have seen in the past, H1N1, H5N1. There's been some cases reported recently in China of H7, H10. And so that's all of us in the field were always thinking it was going to be a new strain of flu, and it was not. And the day after, they told me it's a coronavirus, but it's not SARS, and it's not MERS. But the sequence should be available any day. And on January 11, the sequence of the SARS-CoV-2, as it is called now, the virus was put online. And over the next 2 days, our team worked almost around the clock to figure out how to design a vaccine that they thought could be efficacious against the virus. The piece where we got lucky, and in science, the prepared minds have always an edge, is that we had been working on a MERS vaccine for the last 18 months before the SARS-CoV-2 virus emerged. And so we had tried a lot of different antigen or protein of a virus and to make different vaccines against those. We tried combinations of different viruses. We tried fragment of antigen versus full-length antigen. And what we learned, which was very useful in those 48 hours early January, is that for the MERS, in every case, the best vaccine that we had tried in animal challenge model was always the full-length spike protein, which is around 4.3 kb. It's a very, very big protein. And so we designed the vaccine. Actually, the team designed the vaccine. I should not include myself in a way. The team designed the vaccine and did a couple of checks on everything, and then literally click order on a computer. The sequence went digitally to our factory in Massachusetts, south of Boston. And then two things happened at the same time. We started to make very quickly preclinical material to be tested in animals, which was made in around a week. And we started making clinical grade product, because in the meantime, by talking to Dr. Fauci's team, we had agreed that we were going to make, as fast as we could, clinical grade product. And they were going to run a phase 1 study. At the time, in early January, before this was just going to be an outbreak, like Zika or Ebola or SARS or MERS. And then a week totally changed my thinking. Was in late January, the week of January 20th. I was at the World Economic Forum in Davos, and I had a chance to spend a week with two amazing infectious disease doctors who, because of all the discussions we're having on the outbreak, were sharing with me on their phone several times a day real-time data they were getting from China. And two things were becoming very, very clear. One is R0 was very high. So the virus was spreading very quickly. And two, the death rate was alarming. And in infectious disease, this is the matrix you always plot, is R0 and death rate. And you know most of the viruses where they are. And it just helps you think about, okay, what are we dealing with here? Of course, in the early days, the error bars are huge because all the data is very poor quality because you don't really know what you're dealing with. But it became very clear that this was very, very infectious agent. I had forgotten, where Wuhan is. So I literally got an iPad in one of those discussions with those two scientists from my bag, and I, Google Map, Wuhan, and I see, a big city. And like, geez, it's a very big city. And then I go on Google and I start to look at flights out of Wuhan. And I realized, which I didn't know, is that you had flights going to all the major cities in the world. I learned later that Wuhan was a big industrial, hub, especially in, for example, automobile parts. And so I told the guys, it's too late. It's everywhere. There are too many flights that have left the city. And then in the middle of that week, if you recall, the Chinese government decided to lock down Wuhan, the city. And I don't recall even reading about the city being locked down because of a pathogen agents in the last maybe 100 or 200 years. And I'm like, okay, what does the Chinese government know that we don't know? And between that and the data we had, it became clear it was going to be a pandemic. So I was supposed to go to Germany the next week. And I woke up kind of in sweats on Saturday morning saying, what are you doing? You should not go to Germany. You should go to Washington, D.C. And so on Sunday, I spent the day on the plane from Zurich to Washington, D.C. I bought a one-way ticket. And I spent the whole day in D.C. going to see Dr. Fauci and his team at the NIH early in the morning, going to see the Department of Defense, DARPA, going to see BARDA as part of HHS, and going to FDA to discuss with them, okay, how we're going to do this. And so on March 16, the clinical trial started, which is a record. It took us, 63 days from sequence available to dosing a human with a clinical grade product for phase 1. The phase 2 started in May, and on July 27, we started a 30,000 participant phase 3 in the US. The study is almost fully enrolled. As of last Friday, we had 26,000 participants. And as of last week, we had, I think, 12,000 people who got their prime and their boost shot. So we're almost halfway in terms of boosting the study. As we've said last week, we anticipate to have interim efficacy data in November. It could be October. I think it's unlikely. It is scientifically possible, but I think unlikely. I think November is a good base case scenario. And what we anticipate, assuming positive efficacy data, is to file an emergency use approval to the FDA so that the vaccine could be made available to healthcare workers and or people at very high risk, like the elderly. And again, that will be a determination that we'll do based on the clinical data and talking also, of course, with the FDA. They have, of course, the final say. We have also communicated last week that we are on track to have around 20 million doses available by the end of the year. And for next year, 21, we are on track to deliver at least 500 million doses. And the team is working really hard to try to get to 1 billion doses. It's still too early to know exactly where we're going to land in that, what seems still a large range because of raw materials. We have a space to make the product. We have the equipment. We have almost all the teams hired and trained. That's going to happen. I'm not worried about it. But because the increase of production is so incredible versus what we've done last year, that I'm not yet 100% sure that all of our suppliers will be able to give us on time, every time, the key raw materials we need to make the product. And so, of course, because we're making a medicine, we cannot make the product, if one ingredient is missing from the recipe. We have to follow exactly the protocol of making a high quality product, because of course we're going to be injecting that into real people, and quality cannot be compromised. So there might be days next year where for a day, for 2 days, I hope not more, we're basically going to be shutting down manufacturing with waiting to have the ingredient missing so we can restart making another lot. So it's going to be quite an incredible level of stress and a lot of work for the teams. So that's kind of a bit where we are. Happy to take, of course, question on this. Maybe to close the piece, I hope after this awful pandemic that is taking such a high human toll and a high economic toll. I know so many families have lost jobs, lost income, and are really suffering. I hope that we and the governments around the world learn how to prepare better next time, not only in terms of course, PPE and things like that you've all read, but think about it. If we've had a bigger facility, a bigger factory at Moderna, if we had been working on more viruses and already tried them in the clinic, we could have gone faster. already the timelines we are on right now are incredible. Usually, as you know, it's 4 years, 6 years, 10 years to get a vaccine to the finish line. And we're currently on track, and I have to knock on wood, but we're currently on track to potentially get the phase 3 data before the end of the year, which, if you think about it, a 12-month time frame to do a phase 1, a phase 2, and a phase 3 without cutting any corner on safety, just taking a lot of business risk, a lot of financial risk, with including the help of the government, is just incredible. If you had told me on January 1st, as I wake up, after celebrating the new year with my kids and wife at breakfast, that Stéphane, in a few days, you're going to learn about a new virus that you've never heard of. And by the way, by the end of the year, you're going to have phase 3 data of a vaccine that the team will have gotten through the clinic, through a full phase 1, a full phase 2, and a full phase 3. I would have told you, what are you talking about? It's just impossible. It has never been done before. And the piece that I've been really excited in the last 9 months, despite all of the stress and the long days and the long weeks, is the extraordinary collaboration I have seen between academia and industry, across companies in industry, and with the government. NIAID and Dr. Fauci's team have been amazing, helping us, guiding us, running animal challenge models for us, and so on. The FDA has been remarkable. They've dedicated teams for every company so that we had a full team dedicated to just our vaccine, like we did for Pfizer and Johnson & Johnson and everybody. Instead of taking sometimes days or weeks to come back to companies, usually it has been hours, sometimes 2 or 3 meetings in a day, to make sure that we can save every hour, because at the end of the day, we know every day that we can cut and shave into the development timelines will mean people will not get sick, will mean people will not die. And there's never been a better time to exemplify why what we do as an industry and why it's important to get young adults like you, trained in science, so that kind of this continues to go. So what I think for the future is we could have what I call a never again plant. A plant that is able not to make, a few hundred doses if there is a new pathogen, but could make, 30,000 doses, 1 million doses, 20 million doses. It's just math. Tell me how many doses you want in the first month, and we can do the math backward of how big of a plant we need, how much raw material we need stored in the basement, ready to go if there is a pandemic. So that's a piece I would love to talk with a group if you are interested, is what are the things that we can do as a scientific community so that when another thing like this happens, it could be in a few years, it could be in 50 years or 100 years, when it happens, that because of science and because of collaboration, we can reduce the human impact that has been a tragedy this year. So with this, Jim, I suggest it'd be more interesting if we get into Q&A.
Thank you. Thank you very much, Stéphane. So let us move to the Q&A part of the hour, but I'm going to begin and have the pleasure to introduce the students that will be participating in this. And I'll call upon each one of them. I'll start with Peyton.
Hi there, I'm Peyton. I am a junior bio major from Frankfort, Michigan, and I am part of the Wilson Institute.
Great. We also have Micaela.
Hi, I'm Micaela. I'm a senior from Shelby Township, Michigan. I'm a biology major and a Spanish minor, and I'm also a member of the Wilson Institute.
Jeremy.
Hi, I'm Jeremy. I'm from Highland, Michigan. I'm a junior, biochem major, and also a part of the Wilson Institute.
Fadwa.
Hi, I'm Fadwa. I'm from Grand Rapids, Michigan. I'm a bio major, and I'm also part of the Wison Institute.
And then finally, San.
Hi, my name is San. I'm from Vietnam. I'm a senior this year, double majoring in biochemistry and music, and I'm a part of the Wison Institute of Medicine.
Nice to meet you.
So we have, Stéphane, there are 6 themes that the students want to cover, ending with preparing for the next pandemic. So, but to the students, I think Stéphane covered the first section, which is research process and vaccine development. and actually part of number 2. So if we could start with number 2, and we'll begin with Micaela and then move to number 3. So Micaela, do you want to get started with questions?
Yes. Okay, so mRNA vaccines and technology are still fairly new to the clinical platform, but they show a lot of promising results. So I was wondering that given the diverse applications of this technology, in what direction do you see Moderna focusing their research in the future? And do you anticipate a heavier focus more on things like viruses or other conditions like cancer?
Yeah, it's a great question. So let me tell you where we were before the pandemic started. So we had 6 vaccines in development in infectious disease. We have 5 medicines in immuno-oncology, which is the field of oncology to try to teach your immune system how to go and eat your cancer. We have a very cool medicines, which is for people after a heart attack. As you might know, when somebody has a heart attack, if they survive it, their heart is going to be most of the time damaged, sometimes very damaged because of a lack of oxygen to the heart muscles. And most probably they're going to get heart failure. And so the drug we are developing with the British company AstraZeneca, which is in phase 2 now, is to inject mRNA in people's heart within 48 hours after an infarct of an mRNA coding for the human protein VEGF. And VEGF basically grow new blood vessel. And the idea here, which has shown a lot of promise and published in Nature in pigs, and had a very good phase 1 study. So now it's in phase 2, injected in people with after a heart attack, is basically the idea is, can you grow new blood vessel in somebody's heart to re-oxygenize the heart after heart attack? So it's a bit like a regenerative medicine type of medicine. It has only one intervention, so it's only kind of one intervention. It takes around 10-15 minutes to inject. And then you let basically the human body heal itself. So we need to see in phase 2 if it's going to work or not, but it's very cool. And in the pig model, which in cardiology, as you might know, is very predictive to human, has shown very exciting results. And then is rare genetic disease. And we had a chance to work with Jim and the Institute on rare genetic disease. For example, rare genetic disease in the liver, where kids are missing an intracellular protein in the hepatocyte. And the question here, as we've shown and published in animal models, is can you get the mRNA inside an hepatocyte, bring the instruction for a protein that the kids, because they got the bad DNA from mom and dad, they cannot make, a good protein like we, on this Zoom call can do, and can you restore the function of their liver? The animal models are very exciting, but we need, of course, to see in the clinic. We have a couple green light from the FDA to start clinical trial. And so we're hoping to get that answer very soon. So that's where we were before the pandemic. What we're going to do moving forward is I think we're going to do more vaccines because we have had a very interesting finding recently that we shared a few weeks ago, is that because of all the progress we made in terms of both the science and the manufacturing process of our mRNA, we've shown that in the elderly, we're able to show the same level of neutralizing antibody in their blood than the young person. Usually most technology, if you look at it, whatever you make in a young person, let's say 10 or 20, and in an adult, usually you see half or less than half. And what is really remarkable is in the phase 1 that we shared recently, we had 3 age group, 18 to 55, 56 to 70, and 71 and above. In the 3 age group, all the participants made a very similar level of antibody that were at or above the level of neutralizing antibody that people that had got naturally infected and that convalescent sera were able to demonstrate. So I think more vaccines in cancer. I think we're going to wait to see clinical signal because mRNA is a platform. The application of cancer has not yet been de-risked in the clinic. So we're ready to go with many more drugs in cancer that are in the labs. And we're just waiting to see does our technology work in cancer or not. If it's no, we will assess is there something else you should try, or could you just decide that cancer might be not the best way to use mRNA, at least at this stage of where the science is? Or if we get a positive signal, we'll have many more drugs moving from the lab into the clinic because we have de-risked the technology. The other place where we started to do, where we want to invest a lot, is autoimmune disease. We have 2 interesting new medicines that are getting very close to the clinic. We're going to get clinical signal pretty quickly, and if they are good, given there's still so many unmet medical need in autoimmune disease, I think we're going to go there. The other piece, and I'm going to stop there, is we're also working with Vertex into the lung. You know, Vertex, you might know, is the leading company in cystic fibrosis. So it's a rare disease, but not of the liver, in this case of the lung. The kids are missing an enzyme for having that normal lung functions. And so we are partnering with Vertex to try to see, can we build all the technology and the science to deliver via aerosol through your mouth into your lung mRNA, to be able to get mRNA into the cells of the lung so that you could make the right protein inside the lung. So that's a long answer, sorry, but just a lot of things that we can do.
Great, well, thanks. So students, why don't we go to vaccine effectiveness and safety, and we'll start out with Peyton and then Jeremy and Sam.
So something that we deal with annually is the flu, and having the choice between receiving the influenza vaccine or deciding not to. So my question is the vaccine for COVID-19 shaping up to be a vaccine that will be offered annually, or is it too early to tell within your process?
It's a great question, Peyton, and the short answer is we don't know yet. And we as a collective, we as an industry, this is still a very new virus, as you know. We don't know yet, first, how long the vaccines are going to protect people. We don't know yet if the virus is going to mutate or not. No, it's already mutating. There's an incredible collaboration around the world to share the mutation. So far, they are not in a place that we believe will have an impact on the efficacy of the vaccines, but we need to continue to monitor that carefully. I think the answer also might be different between an older person and a younger adult. And so we just need to see, because the level of antibody might not be predictive of protection of disease, because even if your antibody, of course, over time is going to wane a little bit, what we've seen actually when you give a boost, while it takes for the prime, several weeks for your immune system to mature and to get good neutralizing antibody, when you give a boost, within a couple of days, you have almost a log increase in neutralizing antibody. And if you think about it, the boost is a bit like like mimicking an infection, because you represent this foreign agent into the human body. And so there's a school of thought that you might have, even if you have low antibody level, still the ability to make neutralizing antibody very quickly if you're infected again, where you might still get an infection, but you might not get disease. And that's a piece that we'll need to just understand through clinical study. We're going to monitor, for 2 years, people on the vaccine. We as an industry, and with, of course, the help of the CDC and the FDA, needs to do a lot of epidemiology study to understand duration of vaccines.
Jeremy?
Thank you.
Thanks, Peyton.
Due to the novel nature of COVID-19, you mentioned this will be the fastest vaccine ever developed. What precautions have been taken and what lessons have been learned to prevent a Qatar-style polio incident? In other words, what can we tell our friends and family to reassure them of the safety of this vaccine?
Yes, so at this stage, we do not have the phase 3 safety data because the phase 3 is ongoing. So I want to be extremely cautious because we have only the phase 1 data. So far, the side effects we have seen in the vaccine and that we publish in the New England Journal of Medicine are very typical to what you see in, like, say, a regular approved vaccine. You see, there's 2 types of side effects with vaccine you monitor. One is local side effects, like, pain in your arm being a bit sore, maybe a bit of redness at the site of injection. And then the systemic side effects, like, do you have fever? People have chills, headaches, or other symptoms like that. And of course, more worrying symptoms. So far, what we have seen with this vaccine, but also with the over 9 vaccines that we took to the clinic with this Moderna mRNA technology, is we've seen across the vaccine very similar side effects. And now we have done, I think, around 2,000 people before the phase 3 of the COVID, which I don't have the data for. And what you see is very similar to commercial vaccine. You know, people have pain in their arm for a day or two, which is not surprising. You stick a needle in their muscle, and when you press, you get liquid in their muscle. It doesn't feel good. Even the placebo group, when you look at them, they always complain about the pain, as you would imagine. Some people have a bit of redness around the needle track for a day or two that goes by itself, which, you know, my immunologist kind of like, because it means it's good. There's an immune reaction, and we want to teach the immune system, so that's kind of good. And at the systemic level, what you see so far is some people have a tiny bit of fever that goes away even without taking a Tylenol. Some people have, you know, some chills. A bit like when you get a flu shot, you know, some years I get my flu shot and I feel great. Some years I get my flu shot and that day I feel a bit tired and I go to bed, and the next day I'm fine. Or I have a bit of chills, you know, I go to bed, I feel like I have a bit of a cold, and you go to bed and the next day you're fine. So that's what I've seen so far. With vaccine, what is really hard is you might have a very rare side effect that you need a big N to see. So that's why what the FDA has said for this vaccine is they want a minimum of 30,000 people tested, which is on the mid to high size of what has been done in the past. There has been vaccine approved with, you know, 5,000, 10,000 participants in the phase 3. And the reason they wanted that requirement is because given the pandemic, it was clear that if a vaccine was going to get approved, you might have millions of people getting the shots very quickly after to protect themselves and protect their loved ones. And so it was important to have enough safety database. And so we need to monitor it very closely. As I said, you know, we as an industry and the FDA have not taken safety risk in doing those vaccine studies, you know, phase 1, phase 2, phase 3. As you might have read in the media, there was a vaccine put on clinical hold recently, the vaccine from AstraZeneca, which I think shows that the system works, that if there is something that is questionable, the ethics committee and the safety board that are independent from the company, they are a group of clinicians that are used to do clinical research, that are not made of any employees from the company, totally independent from all the hospital and academic centers who run the studies for us. They look at the data and they are the one who decide. So it's not like if something strange happened, they call the company and say, this happened, what do you think we should do? No, it doesn't work like that. They will call us and say, oh, we're just letting you know we're putting the study on hold. And it's a very important kind of check and balance to ensure that the safety of a participant is priority number one, so that those clinicians and ethicists can look at the data and make a determination. Could it be caused by the vaccine or not? Like I know there was a case in the AstraZeneca vaccine described in July in one of the earlier studies in the vaccine where somebody developed MS and they put the study on hold. And when they did the clinical assessment of that patient, they realized that it's somebody that had not been diagnosed, but given the stage of a MS disease, it couldn't have been caused, you know, a week before by the vaccine. Just didn't make in their clinical judgment any sense that this was the case. So after finishing the analysis of the clinical case, they decided it was not related to a vaccine, and they restarted enrolling people and continuing the study. And so this is typical in clinical development. I think this one, because we're all in the middle of a once-in-a-century pandemic, and because of the impact on all our lives, there is incredible scrutiny on the vaccine development. But for me, who has been in the life science industry for 25 years, this is just how things are run. But I think there's a lot of education and communication to do. One of the things Moderna has done last week, which I'm very proud that the company did that, is we put online the full phase 3 clinical study protocol. Usually this is never done while the study runs. And we put it online because we say, look, we owe it to the scientific and clinical community, we owe it to potential future people who want vaccination, that everybody sees a fully unredacted version of a protocol so people could make their own assessment. Because what we want is, the team has been working 9 months, 7 days a week to save every day for the approval of this vaccine. if, you know, a lot of people do not believe in the safety of a vaccine, that would be terrible for us, as you can imagine. We sacrifice so much to try to help, you know, people with making a vaccine available to stop this awful virus. And so the transparency and people knowing that we care deeply about the safety of the vaccines is extremely important to all of us.
Great. Why don't we move to the role of government? And I think we have 3 questions here that I think we'll get through: Micaela, Fadwa, and San, but we'll start with Micaela.
So scientists have been predicting a large viral outbreak, such as the COVID-19 pandemic, for years, but funding in this area has been very low. So I was wondering that now that a pandemic level viral outbreak has occurred, do you predict any changes in research in the future? And do you think there will be a heavier focus on funding for producing preventative treatments?
It's a great question. So I'm going to be cautious with predicting because sometimes only fools predict. I'm going to tell you that I'm hoping, and I'm going to work really hard here on my commitment to engage with governments around the world to fund more research around emerging pathogens. One of the reasons we've been able to move fast is because, as I told you, we have been working on the MERS vaccine before SARS-CoV-2 happened. There's around 10 to 20 viruses that the scientific community think are at a high risk of creating more outbreaks. And what we should do for all of those is do all the preclinical work to test, as we did with MERS, you know, which protein of the virus will give you the best vaccine. And then you should take those into phase 1 and phase 2, so that if a mutation of one of those viruses emerges and becomes an outbreak, you can very quickly, through the sequence of a virus, design a vaccine in a few days like we did. But instead of going to a phase 1 and then a phase 2 and then a phase 3, go straight into a phase 3 with 30 or 100,000 people. You could also make the vaccine right away. And once, let's say, an mRNA vaccine has been approved, potentially make it available to healthcare workers as soon as you know the vaccine is working. So I think there's a lot of things we can do. I've had the chance through the last few months to talk to around, 10 presidents or head of states across the world. And I've told all of them the same thing. You are spending billions of dollars a year just to maintain nuclear weapons. And you're not spending a couple $100 million or $1 billion or $2 billion a year as a coalition of governments to make sure that we are ready, that we do all the work that will save precious days, that will save many, many lives. And it makes no sense. It's crazy we're working on our heads. And so one of the things I definitely, as I talked to you at the end of my remarks with these never again plans that I want to build, is I really want to, as soon as our vaccine launches, hopefully in the next few months, I want to really focus my energy on how do we make sure that such a human tragedy never happens again.
So again, rolling on the role of government has come up in the past, and Fadwa, you had a question on that, and San can follow up. The role of government.
Yes, so I noticed that government officials across the country have stepped up to speak out about the pandemic, with some advocating for a quick fix and others stressing the need for stricter guidelines and more time for testing vaccines. So what, if any, government influences and pressure has your company felt while researching vaccines?
Yeah, it's a great question, and I get it a lot, as you can imagine, reading the headlines in the news. And I can tell you, I've actually got no pressure since early January by any governments in the world. I've had no phone calls or emails or meetings where a head of state in any country has told me, Mr. Bancel, we need you to go faster, we need you to cut corners. What I've had is actually a lot of help. As I said, the FDA and their teams have done a remarkable job to help us not cut corners, but figure out how to go faster with the same level of scientific and clinical rigor. BARDA, which is part of HHS, has helped us by allowing us to get funding so that we could do things in parallel and take a lot of business risk that would have been really hard for a company like us that has never turned a profit to take. We are now making product, assuming success, in our factories. We had to buy hundreds of millions of dollars of machines, hiring hundreds of people, and we're making as much vaccine as we can make, because think about how it will be a human tragedy if in a few months, if the clinical data supports it, we could have a vaccine approved, and I will be on TV or my team telling the world, well, now you have to wait for 6 months because we need to start making products. You know, people will feel so let down by this outcome. And so we said we have to make products at risk now. There is no other way to do it from an ethical standpoint. But then the question is, where do you get the money to do that? And this is where governments have been very helpful, not only in the US, but around the world. We've got already quite a number of orders that have been placed for our vaccines by governments so that they can give it to their populations. And what they have done is they have paid us upfront so that we could turn around and give the money to the suppliers to get more raw material to make more vaccines for more countries. So I have to say, even though not everything has been perfect, I feel really good about where we are now in terms of the help we've gotten from government. And as I said, we've gotten no pressure to this date, despite all the things I've seen like you in the media. I can tell you, and I've talked to other CEOs in the pharmaceutical industries making vaccine, and I've heard the same message as the one I've given you, which is my colleagues who run other companies have had no pressure on going faster because it's very important for all of us to do things with the highest scientific and clinical rigor, and always with the safety of the participants in the studies in mind. Those are, you know, parents, those are brothers and sisters, those are kids of real people, and we cannot cut corners and put their safety at risk.
Sam, you had a question in this area.
Oh, yes. So recently I saw a town hall event hosted by ABC News in Philadelphia, where President Trump was invited to talk about, to talk to like undecided voters about the pandemic and things like that. And I saw that he gave out a misinformation that the pandemic would go away even without the vaccine with time, because people would acquire herd immunity. But he actually said herd mentality. But then on the other hand, Trump keeps telling the press that there will be a vaccine by the end of this year. So how do you think Trump's inconsistency about the severity of the pandemic impacts the credibility of a COVID-19 vaccine for the American people? Like, do you think people would trust it? Because, because even though the current administration is giving people mixed feeling about it.
Yeah, it's a good and important question. One of the things the industry did 2 weeks ago is the CEOs of all the companies that are in the clinic, signed a joint pledge, which I don't recall in 25 years in this industry happened, that we will not submit to the FDA any file to get approval until there's a complete data set for safety and efficacy. And the reason it's important because one other way you could have had some vaccines approved is by submitting neutralizing antibody data, which we already have for our vaccines. We don't have a big safety database yet, but we have neutralizing antibody data. And while for some of our viruses like flu, it is scientifically appropriate to give approval based on neutralizing antibodies because there's so much understanding of the virus and the disease that we believe at the scientific level, we understand how much antibodies is protective of disease. For SARS-CoV-2, we don't know where it is. We have some indication with convalescent sera, but there are so many things that we cannot explain yet scientifically about the SARS-CoV-2 virus and manifestation or not of disease, that we need to run efficacy studies where you do placebo-controlled in a large data set to understand in different population by age group, by comorbidity like diabetes, like overweight, like cholesterol, high cholesterol, but also by ethnicity. We don't know yet, but we will know soon. For African American, for people with Asian background or Latino background, how does the vaccine work for them, both from safety and for efficacy? And so This is all happening now. And so one of the things the industry did is I've signed, with my name, it has been published in major papers, that I will not submit to the FDA for an approval of our vaccine until our scientists and our clinician and myself feel confident we have the total package that we will give this vaccine to our children, our parents. And that's really how I set the bar myself. As I've said before, once the vaccine is approved and I'm allowed to take it, because today I'm not allowed to take the vaccine. If not, I would already have taken it. Because it is unethical for somebody who works in a company to try a product in clinical trial, because people are worried that I will not report the side effects because I'm conflicted being working for the company. But as soon as I'm allowed by law to take the vaccine, I'll be the first one to show my arm, and I will have my wife next to me and my 18-year-old daughter next to my wife. And I'll go first, of course. This is just how confident we are. That's the bar we're setting, as you know, because many of you, are studying science and medicine. We came to this industry to help other human beings. We have worked so hard for the last 10 years to make the science work, and in the last 9 months non-stop, including Saturdays, Sundays, we have pulled all-nighters, because every hour matters, because we know somebody is going to get hurt if we don't do the best job we can. And what we've tried to do is to always make sure that quality and safety is not compromised. We have wasted money, and I've told our shareholder, we made mistake of overspending on some things. But the piece I can tell you we have not done is cut corner on quality or safety, because this vaccine could be injected in somebody we love, and that's always the mindset we have. That's the acid test, is will you be willing to inject this product to somebody you love?
Probably time for 2 last questions. Peyton, maybe you could ask your question about research funding and early on.
So when Moderna was starting the process of coming out with the vaccine, were you receiving government funding, private funding, or both? And were you receiving the amount of support you needed to pursue this?
That's a great question. So in the early days, we got very help because the NIH, Dr. Fauci team committed right away to pay for the phase 1 study, meaning we didn't have to take the risk and pay for it. I got a foundation called cphi, which is in charge of helping prepare for pandemics, which was started by the Wellcome Trust Foundation and the Gates Foundation, gave us the money to make the product and to pay for the manufacturing of a phase 1 product. And so that was very helpful. The phase 2 material, we paid ourselves with our shareholder money, because I've been asking a few organizations and foundations and I didn't get the money. And I told the board, we have to go and spend our own money because we cannot waste any day. It's just not ethical to waste time. That amount of money is going to hurt us. It's not going to kill us. So let's spend the money. Later on, we got the support for the phase 3 by the US government, which was very helpful. The place where I wished I could have had more help, and we wasted maybe a week or 2 in making more products, is I tried to talk to a lot of governments to help us, give us money to make product at risk, buy machine, buy raw materials. It was back in April, early May, and it was very difficult because at the time we had no human data. And people were very worried for a company that is a new technology with no human data, despite we had made 9 vaccines before where there's a lot of human data. But a lot of politicians do not understand science, and even though you explain to them that if it works for Zika, it should work for this because you just changed the sequence, or if it works for MERS in animal, it should work for the SARS-CoV-2 because it's the same virus, just change a few nucleotides. And so I was getting really frustrated. And one day I was super frustrated to the point that I went to my boss and said, we need to raise more money. And we did something quite out there, which is we raised on May 18, $1.3 billion on the NASDAQ that evening after the market closed at 4 p.m. And we told investors, all of that money is to make the SARS-CoV-2 vaccine. We're going to buy machines, we're going to hire people, we're going to buy raw material. But if we don't spend the money now, which of course we didn't have in the bank because we were not planning for a pandemic, if we don't spend the money now, if a vaccine is approved at the end of the year, we will have no vaccine to provide the population. And I told them that will be just such a waste of life. It would be so unethical that we have to go now. I'm not getting it from government, so it's actually our shareholders or investors. We took the risk. At the time, we've only data in 8 human beings in phase 1 to go and give us $1.3 billion just to go make product at risk, knowing that if a product was going to fail, all that vaccine was going to go into the garbage.
Last question. I think we're going to end where Stéphane, you ended, and that has to do with preparing for the next one. And Fadwa, I'll give you the last question here.
All right, so this pandemic took many people by surprise, and most were not prepared for its severity or its duration. So looking back, what steps do you think were most effective in combating the virus? And looking forward, what can help our global community to better prepare for a similar situation with a new disease?
Yes, a great question. So I think, as you all know, testing is critical because as scientists, if you don't know what you're dealing with, if you don't know who's sick and who's not sick, you cannot operate. And so I think making sure we have all the latest technology, that we are ready, that there is machine in all the key labs that could be put active very quickly is critical. And you've seen the countries that have been the best at testing have been able to control this very quickly. The other piece is, as we see now, mask. This is a respiratory disease. So I wish we had been wearing masks earlier. I think we could have saved a lot of lives by wearing masks earlier because this thing spreads through the respiratory system. And so are masks perfect? No. But do masks reduce your chance to get sick or your chance if you are sick and you're asymptomatic and you don't know it to make other people sick? I think it's important. And then I think it's on the science, as we discussed, investing on those most important or more risky 20 viruses and doing all the preclinical work, doing phase 1, phase 2 to de-risk and start getting the process moving. I mean, think about this. If we had been able to get funding to do the preclinical work on MERS, which we did, and then if we had run a phase 1 and a phase 2 on MERS, which we did not, we could have started a phase 3 study in March. We could have had the phase 3 study done in June, July. So it means we could have had the vaccine this summer. We've been losing 600 to 1,000 Americans, and I'm not even counting the lives in the other countries, every day. That is why we need to make sure that the lessons of this pandemic are not going to waste, and that as soon as this one is in control, that we go and we invest, because it's not at the end of the day a lot of money versus just what we spend in the military. Go and do the science correctly. Go and have stockpiling of tests, of masks, and so on, PPEs and so on for the clinical professional and so on, to make sure also that those stocks are replenished with, because everything has a shelf life, so that whenever there is another outbreak, we can react much better and we lose more lives and we have less people sick.
I want to thank the students for participating in this webinar, and Stéphane, you and your colleagues. I mean, it really is a story of commitment that you've had to make, but leadership, going at risk financially at such a significant amount really is a kind of risk we need to take. And I think the biopharmaceutical industry hopefully will be viewed in a different light nowadays, in light of how you guys have come together to try to deal with this global crisis. And all we can say is we're with you, and Godspeed. We hope things work out. And thank you again for sharing your story personally and directly with those who see this or are going to view it by YouTube.
Great. Well, again, thank you so much for having me tonight. It was great to be with all of you. Keep studying science and medicine. We need a lot of smart young scientists and doctors to get the place to a better world. There has never been, I think, a better time, even before COVID, to get into science. This is a century of biology. We can do so much for medicine, but also for the environment and so many other pieces of our life through science. So stay healthy, everybody. Wear your mask, take care of each other, study hard, and I look forward to meeting some of you in person at the next occasion once we will get a safe and efficacious vaccine. So thanks, everybody, and stay well.
Great. Thank you all. And everyone have a good evening. Thank you very much.
Thank you so much.
Thank you.